Well, as you see here, we are at the campus of University of Louisiana at Lafayette, and we are here in the biology department. That's where I'm a professor. And what is great about this biology department is that it's very strong in biodiversity, the study of biodiversity, especially marine biodiversity. So I am studying marine algae, or the seaweeds, in the Gulf of Mexico, and then my colleague, who you're going to interview a little bit later, studies crustaceans, decapod crustaceans. So we really cover basically all the invertebrates and the seaweeds that are really at the base of the whole Gulf of Mexico diversity. So it's a very good place to do this type of work.
Okay.
Well, for the rapid funding, we are trying to continue what we did before, is basically recollect all the organisms at a special depth in the Gulf of Mexico, close to the depth where the oil spill may have an effect. So we are actually looking at recollecting previous expeditions we went to of algae and invertebrates. So more particularly the macroalgae, what we call the seaweeds, and then Dr. Felder is going to talk about the crustaceans and mollusks and so we collect. So what is very interesting is that, in the Gulf of Mexico you have all those hard banks.
What are the hard banks?
Well, the hard banks are those hard substrata, and the ones that are close to the coast of Louisiana offshore are basically salt domes. So what is very interesting is that those salt domes trap all those hydrocarbons. So there is a lot of oil and in there, but outside of them you can see a whole bunch of organisms that grow on top of those salt domes. And a lot of those algae that are dominant are, of course, the seaweeds, which are, of course, one of the I'm biased, but one of the most important organisms in the ocean, because they are the primary producers. They are the one that produce the oxygen that enables all the other organisms to live. So if there is something wrong with those seaweeds, and they are the base of the food chain, everything else can fall apart, you see. So even though they may look all right now, we're going to go in December, we're going to recollect there, and we're going to see what the effects are, if we see any effects right now. Maybe in December, everything is going to look fine, we don't know. What's going to be important is to look in the long run, what the effects are going to be. How the algae, will they be able to reproduce, etc., how healthy they are. Which one are going to have disappeared because of the oil spill? Which one came in as a result of the oil spill? We don't know yet, you see. Was basically to recollect in the same places we have collected before in the Gulf of Mexico. In 2003, Dr. Felder and I, we were awarded the NSF Biodiversity Survey and Inventory Grant to actually collect all the seaweeds and the invertebrates throughout the Gulf of Mexico. And so not only in the northwestern Gulf, like where we are here, but also in the southeastern, southwestern, and northeastern Gulf of Mexico. So over a couple of years, we basically collected throughout the Gulf of Mexico, around the depths between 45 to 90 meters depth. And Dr. Felder also went a bit deeper because a lot of his organisms live deeper. But the interesting thing was to collect those organisms on those hard banks. And those hard banks are going to be different, whether it's in the northwestern Gulf, like here the salt domes, or the southeastern Gulf and the southwestern Gulf, where you have all those carbonate rock formations. So what we wanted to do is collect everything we had, because the region is not so well known. You have to really go deep to find interesting organisms. Because, of course, the coast of Louisiana is not very interesting for my type of organism, the algae, the seaweeds, because there's too much the influence of the Mississippi River. So to really get the truly marine organisms, we have to go offshore and very deep. Well, not very deep, not like thousands of meters, but like in the medium depth range, like between 50 and 90 meters. And that's where we found our very interesting organisms. And what we found is that it's like a gold mine in the Gulf. Like I've been all over the world, but I think one of the areas I like most of all is offshore, the Gulf of Mexico, because you have really very interesting assemblages of algae that you do not find anywhere else. So it's like preserved from historic times, and this is basically maintained here in the Gulf of Mexico. So we have like families and orders of red algae, marine seaweeds, that you basically only find clustered here in the Gulf of Mexico together. So it's very, very interesting how it got basically preserved.
Why is this preserved here?
Well, they came here from millions of years ago. But with in some type of families that is preserved in also the Mediterranean Sea, the South Australia. So with the historical biogeography, what we're trying to do is seeing what are all the different pathways of how those groups of algae got into the Gulf of Mexico. And we see all different pathways, more recent ones, all distributions. So we try to figure out what are those algae of the Gulf related to. But before you can say that, you need to know what it is. So we look at those seaweeds based on both morphological and molecular evidence. So for morphology, we look at the reproductive stages, the vegetative stages. We compare with other algae from other parts of the Gulf and parts of the world, because we cannot really look at the Gulf by itself. We have to look at the organism of the Gulf in relation to the seaweeds from the Caribbean, the Atlantic, the Pacific, to really try to understand basically the whole biogeography of groups of marine algae and other organisms. So, but what is so interesting with the Gulf is that you can learn so much from this area to understand the algae from all over the place, you see? That's what we try to do.
What's interesting around the Gulf?
Well, we might see a shift in the number of species, in the type of species we have seen, in the reproductive status, how healthy they are. Maybe we may see some species that are there, but who knows, maybe the effects of the oil and the dispersants affected their reproductive status. So they may maybe not reproduce. They may not produce spores. We don't know yet. See, the algae, they cannot just walk away. They are stuck. They are marine plants. They are stuck to the substratum. So it's not like a fish that can basically swim away. So we don't know the effects of it. So that's why we're going back in early December and then in April to see whether there has been a shift, or what basically was unaffected by the oil spill, what may be affected, and how quickly it can recover the species that we didn't see in one time frame. Well, what we will be doing when we go in December is collecting our specimens, the seaweeds and the invertebrates, by dredging, by small dredge, and then we bring it on board, and then we sort it, and in the lab we identify it, analyze it, we look at morphology, and also we are going to basically extract the DNA from it to do phylogenetic studies, evolutionary studies, molecular studies. And then we can see basically how the algae, are they the same that we found before? Because they may start looking a bit differently. We don't know yet. They are kind of sometimes very fluid looking organisms. So we want to see what is there now and compare it to what was there before the oil spill. And we may have to look at those things for years to come, because maybe the algae might be there in December or in April, but how healthy are they? You see, we don't know if other algae are going to be able to take over the place of some algae that got wiped out. And maybe some algae were there in the spore stage in the Gulf, and somehow they found a new niche and are taking over the place of algae that went there before. So we don't know all those things. It's all we can say. It's going to be a very long-term project. Well, we think that, I think especially for the marine algae, that there may be a shift in the composition, because we already looked at the effect near shore. Because we went there before the oil spill near Port Fourchon in Louisiana, and we looked at some of the little rocky outcroppings there. And when we were there first, everything looked healthy, like when we went there in May. But then when we went back in July, we saw already a different shift. There was some more blue-green bacteria that took over the place where normally there should have been some very fine little red algae. So superficially, everything looks okay. You see things that look like algae, which are actually bacteria, some photosynthetic bacteria, but they completely took over the site where red algae were before. So that's why we really need to know and precisely what is the composition, because you may look, who knows, maybe they may become invasive species, may come in there. But if you cannot distinguish one algae from another, you're not going to see a difference. So we are very much collections based, so we will be able to really compare what's there now and what was there before, and what's going to be coming there in the future. So we think there's going to be a shift. We think there's going to be some type of effect, but we don't know exactly which one. To look at the biodiversity of the Gulf, because it's a region that's not so well known. And we all know much more about what's going on in the Caribbean or in the Atlantic. Because the Gulf is a difficult place to collect. You have those modern-nose coral reefs, for example, in the flower gardens, of the offshore Texas, between Texas and Louisiana. Well, before you get to the top of those coral reefs, you need to go like 60 feet, let's say. Whereas in the Caribbean, those safe coral heads would be maybe at 10 feet. You see what I mean? So everything is much deeper. So it's difficult to just go there. You cannot just jump in the water and collect all the algae. We cannot do that. So it takes a long time to try to get those expeditions launched. We do it typically with the research vessel, the Pelican, that basically launches out of Lumcon and Cocodrie or Chauvin, Louisiana. So we've been using this boat for years, and it's perfect to suit our collection methods. It's like, I think that's why NSF funded us, because we had that great database that we have been able to accumulate for years and years and years. So now we have something we can compare with. Because otherwise we would know I think they would not have funded us if we didn't have that database to compare collections we are going to do now. And then we can really see what has changed, how it has changed. Well, I think there might be some changes in biodiversity, but it depends on what type of algae groups. Some algae, I think, are not going to be affected whatsoever. Some are tough, but others will be affected. But the problem is we don't know yet which ones. You see, that we're going to have to see in long term. Other algae may come in, maybe like invasive. Well, the Gulf hypoxia is more the effect of all the nitrates and the phosphate that got flushed in, through the Mississippi River. And what that causes, of course, is a bloom of algae, seaweeds and macroalgae. And what happens then is a lot of other animals and little animals, they're going to feed off all those proliferating algae because they love all those nitrates. Those algae love it. But then, of course, at one point they're going to produce the oxygen, but then they're going to die off. And then the dying off is going to consume oxygen. So by consuming it, all those bacteria are going to basically consume the oxygen by decomposing the algae. And this is what, of course, gives you that big dead zone. So what happens now is we don't know if there is some pockets of that has been deprived of oxygen, because we don't know the effects of all the oil and the dispersants, what it does to the seaweeds, because seaweeds are the primary producers. So they need to have all the correct light composition, and so to undergo photosynthesis. And if something goes wrong that we cannot even see, they might block photosynthesis. So oxygen may be blocked. You see what I mean? So this oil spill indirectly can have a big effect on the amount of oxygen in the ocean. And if there's something wrong there, then of course the whole rest of the food chain is going to be affected. You see?
We might be able to see that from what Dr. Felder is doing as well.
Yeah, but he works on consumers. I work on primary producers. So if something goes wrong with the primary producers, everything else breaks down. And we don't know yet. See, it might not happen right away. It might happen in several generations. You see, we don't know what it all does into the oil spill and the dispersants in the physiology of those algae and those seaweeds. When I talk about seaweeds, those are the macroalgae, the big ones. And then we have the phytoplankton, which are the tiny unicellular algae. and both of them produce a lot of oxygen. And if, like I say, if something goes wrong with those seaweeds and those algae, then everything else is going to be affected by it. And so a lot of people don't really appreciate the role those seaweeds do. But like I say, they are probably the most important creatures and organisms within a marine ecosystem, just as they are in the freshwater ecosystem. Because the whole ecosystems that is oxygen dependent is going to be affected by what happens with those seaweeds. Yeah, well, they say with global climate change and the acidification of the ocean, that's going to be very important, because what are coral reefs? It's all calcium carbonate. And when we look at the substratum, when we collect the algae on the salt domes, it's also those little calcium carbonate nodules, what they call the rhodoliths, the little red stones. So those are naturally occurring calcium carbonate nodules, and then a whole bunch of algae are basically covering it. So if there's something wrong with those calcium carbonate nodules, with acidification, you're going to produce less calcium carbonate, because it's going to dissolve with the acid. Then what's going to happen? All those algae will not have all that substratum to settle on. And if they cannot settle on, they cannot survive, because they cannot just walk away. So there again, if something goes wrong with those algae, they don't have substratum because of the acidification of the coralline of some algae as substratum, and also as seaweeds, because a lot of those coralline algae that basically cement all the reefs, and they have all that calcium carbonate in them. So if something, the acidification breaks down all that calcium carbonate, then the whole reef system is going to break down too. So there again, the algae are so important at all different levels, as primary producers, as cementing substratum, as shelter, being shelter for a whole bunch of organisms, little crustaceans, whole bunch of invertebrates, fish. They can all basically grow on and below and on top of all those seaweeds. So it's basically like a little miniature ecosystem that we see like in the seaweeds that are really, well, I'm biased of course, but they are really so important. And not enough people talk about that. With the oil spill, of course, you see what's terrible to see those pelicans. In the news, you see those porpoises. You see all those kind of animals that are affected, that you can see with your eye. But inherently, it's what happens with those algae, I think, effects of them that are going to be really, really important. And it's at those depths between like 50 and 90 meters, because most of the time what we see, what we heard in the news, it's very shallow, and what happens near shore or in the marshes, and very, very deep, where all the plumes are and the bacteria are. But the kind of area between 50 and 90 feet, this is where all that biodiversity is, and that's what we are studying. So most of the algae, people think they only grow on the surface. They float on the surface, because that's what you see. When you go in the Gulf, you go off Mississippi, Louisiana, you see all those brown seaweeds, sargassum weed, sargasso. But this is only a tiny fraction. Most of the algae are attached at the bottom. They cannot move. So they are stuck there. And there is that great diversity. We have hundreds of species of different algae that you don't see from top of the boat. It's all very deep, and that's where we have all those different species, and that's why it's so rich. So people might say, well, we don't see the algae, there's no problem. Well, yeah, it's a problem, because it's what we don't see that's probably more important than what we see. We can deal with things we see. So you talked about what we see on the surface is that sargassum seaweed, and it's very important too, because it's like rafting islands basically, and in those islands you have all little shrimp and little crabs and little young sea turtles who are resting there, who live around the seaweeds. And what we saw, of course, with the oil spill, all of those sargassum weed, it all came on shore on the beach, what it does normally, but it was coated in oil. And that means that all the little animals that grow around that seaweed died also. You see? But this is like just like little one drop in the bucket. That's what we see, we can deal with. What we do not see is what's deep in the ocean, what's deep there on the hard banks, on the salt domes, on the little substrata. What we see with those brown algae and those sargassum is the tip of the iceberg. But it's just a fraction of the whole diversity of the seaweeds in the Gulf of Mexico. And what we are studying is everything else that's attached at the bottom. And that's so important to provide oxygen, to provide shelter, to provide a whole substratum for the whole bunch of all the organisms, for fish, for crustaceans, for everything else. Everything is interconnected. That's why we cannot just look at the algae. If we want to understand the diversity in the Gulf, we have to see what are the algae providing as shelter and as food to a whole bunch of other organisms, you see, because everything is interconnected. And I'm starting with the basis, the seaweed. So if something goes wrong with the seaweed, all the other interconnections are going to fall apart and break down. And then we're not going to have all that diversity and later on. Probably is how the long-term effects are going to be. Because right now everything might look healthy, might look normal, but we don't know in several generations of seaweeds, several generations of animals, what is the effect going to be of the oil spill. It's not enough to say, oh, we don't see the oil, everything is okay. No, it might have very long-term lingering effects, and that's what we try to see and understand. So if in case there is another oil spill, we know what to expect and how to act upon it. Well, we were very lucky, Dr. Felder and I, that we received one of those NSF rapid grants. NSF stands for National Science Foundation, and the rapid grants are typically being awarded when there is a very short-term catastrophic events to study it. So, for example, oil spill or an what else? How do you call it? Well, how do you call this? Something dramatic happens, then they award some of those NSF grants to quickly study the whole phenomenon of it. So that's what we're very lucky to have received.